Hello, everyone. Welcome to the lecture 3 of Introduction to Biomedical Science. This is Hambam Park. Today we'll learn about protein databases. As I announced before, today's lecture will be done through this recorded video. As we learned before, today we are going to learn about protein databases, and we'll cover 4 different types of materials. We'll start from protein classification, followed by protein data bank, or shortly PDB. Then we will learn AlphaFold and AlphaFold database. And at the end, I'll add one more short demo on how to use PDB and AlphaFold DB for your research. Let's start with the first protein classification part. Before starting, I want to mention and recommend you to go to advanced materials if you're more interested in this subject, because of time limit, I'm not gonna teach too much on this part, but it has a long history and then lots of materials you can refer to. So if you want to learn more in depth, then please refer to bioinformatics course, which is covered for a full semester, or refer to textbooks such as structural bioinformatics, shown in the right side. There are 2 ways of classifying proteins. There is as you learned before, you can represent proteins either in structure or in sequence. And then they are a one-to-one correspondence to each other. And then if you classify protein based on structure, you follow this arrow here, and then you search and compare against the structural database and get what class your protein structure belongs to. That's how you get the protein structure class. And then if you grab the sequence, then here you compare against sequence database. By comparing against that, you'll get the protein sequence-based class. In this lecture, we are gonna go individually, starting from sequence database first. First, let's learn how to classify using protein sequence. There are many benefits of using sequence rather than structure for classifying proteins. First off, there are far more sequences of proteins than their structures. If you count the number of entries in the database, the structural database only contains less than 100,000. But if you count the number of sequences, then there are more than 3.6 million. And then because of next generation sequencing, most of the proteins you know have their sequence annotations, but only a fraction of proteins has their structures. So this is why you use sequence for classifying proteins. But because sequence structure function relationship that we learned in previous lectures, sequence alone can be used to tell what protein this is and what function it does. So sequence-based classification is still a very useful tool, even though you don't know the structure. And in this classification, the basic unit for identifying class is sequence similarity. So by definition, classification means grouping. And in order to group, you have to compare between 2 items. And comparison is made by what is called sequence similarity in protein sequence space. So let's say there are 2 sequences like having 4 amino acids like this. One is EKIF, the other is DRLF. And if you just count identical amino acid pairs that match each other, there's only one, phenylalanine here. So in terms of identity, it's just 25%. But you know that this E and D, the glutamate and aspartate, are very similar. Lysines and arginines are very similar. And isoleucines and leucines are also very similar. So people collected protein statistics and grouped which amino acids are similar to each other. And then there's a matrix called sequence similarity matrices, such as Blossom 62, which tells which sequence is similar to others. So if they belong to the same group here, you can consider them as similar, although they are not identical. So based on this rule, you can say these are only 25% identical, but 100% similar. So we're going to use the similarity metric for comparing between sequences and to classify proteins. And then what we need is sequence alignment. Because protein sequences are not in the same length, if you just put in just a simple way, then you can see this kind of alignment to each other. But obviously you can find that E and D match to each other, V to V match to other, D and D to each other. So if you want to insert one gap here and slide whole this part, then you'll get much better alignment to each other. So this logic is called sequence alignment. And this gives much better view on how similar two sequences are to each other. And this is well-established computational algorithm that you don't have to bother. You can use just computer, web servers, or programs to get the proper alignment to each other. You can also do this for more than two proteins, and that is called multiple sequence alignment. You can align related proteins in a single page like this, each column containing similar amino acids. And you can see that some columns are similar, some are identical, and some are unrelated. By using this multiple sequence alignment, you can collect the information which residue is important for that protein family and which are just random. And then you can easily imagine that the residues that are highly conserved in identity or similarity, they will be important for the protein stability or function. These are two main factors why protein sequences are conserved. And then we can construct what is called sequence profile. If you make an MSA, then count the numbers of sequences or amino acid that appears for that column. You can get this kind of logo showing which amino acid is more conserved at that column through that family. So based on this, you can represent the entire protein family and then important residue points. And this sequence profile becomes the beginning point of protein classification. By using this sequence profile, you can cluster proteins into different hierarchy. PFAM is the most well-known database of sequence-based protein classification, and proteins that are sequentially similar are grouped as families, and then their further relationship is grouped by similarity in their sequence profiles. You can access to the PFAM definition of proteins by clicking into this website. There are about 22K PFAMs known up to date. And each protein has their own PFAM annotation shown here. For example, this protein called USP7 belongs to PFAM 443. And then all these proteins that belong to the same PFAM are similar in their sequences to each other. Let's move on to next part, classification by structure. Before beginning, we have to define a concept called domain. Preston domain is the main classification unit when you do structure-based classification. The domain means self-stabilizing and folding unit of protein. So this is not always equivalent to protein chains because single chain can be more than multiple domains, or more than one chain can form a single domain. So here is an example. This is a single protein chain of pyruvate kinase. So you can easily see that there are three parts that you can easily understand they fold by themselves. And then they are just connected through linkers, shown here. And then we can call this protein has three domains. And then classification is not made on this protein level, but in domain level. Like when you ask what structural class this protein belongs to, then each domain has different classifications. There are two popular structure-based protein classification databases. One is called CATH, and the other is SCOP. And then the common thing is that they both take the domain level grouping of protein strategy. But then because their domain definitions are different and their logic for classifying protein structures are different, they have different annotations. But if you go to each protein's page, you can easily get both information at the same time. For example, the protein we looked at, USB7, you can easily get the SCOP classification. Here it says it's alpha and beta protein in class level, and in fold level or superfamily level, it belongs to system proteinase. If you go to CATH definition, it's defined as mainly beta, and it's a single shoot architecture, and it belongs to ubiquitin family, deubiquination enzyme superfamily. So they have different classification logic and definitions. These two definitions are used as mixture, and main reason for that is because this structure-based sequence structure classification is quite subjective. So it's better to have both definitions so that can complement each other. We'll look at how CATH and SCOP definitions are made in the following slides. First, CATH. What CATH stands for is how the data structure is organized. It stands for class, architecture, topology, and homology. And it's actually hierarchical classification logic that the biggest group start with a class, more detailed one followed by architecture, and even more detailed using topology and homology. So class defines what the general secondary structures are, as shown here. The architecture deals with their numbers and orientations, and topology deals with the actual detailed shape. If you access to this sequence website, you can get many useful information by clicking this many structure-based analysis options. The next is SCOP. You can also access to this website through this link. It stands for structural classification of proteins. It has very similar definition as CAS, but their names are different. It's also hierarchically defined. The biggest category start with class. And it's same, all alpha, all beta, alpha beta, or alpha plus beta. And the last one is disordered. And followed by fold level, superfamily level, and family level. Here, the fold almost corresponds to topology level in Cas, by the way. And then you can also access to this website to search the protein scope classification or download whole protein structure that belongs to specific class or specific fold or superfamily. To summarize what we learned in this section, proteins are classified by sequence or structures. And in the structure-based classification, we used protein domains as units. Protein domain means, again, self-stabilizing, self-folding protein piece. And we learned that the structure-based classifications are built and organized in a hierarchical way, starting from broader definition to detailed definitions. So each protein having 4 leveled definitions, starting from class to lowest level. And you can easily access to these annotations of your protein by going to one of these websites. You can get all this information, what your protein belongs to, which class your protein belongs to.